Are we talking mainly
Yes, yeah, get started. It's my great pleasure to introduce our speaker today, Dr. Luke O'Neill from Trinity College. Luke received his BA from Trinity College in Dublin and his PhD from University of London. He's currently professor of biochemistry in the School of Biochemistry and Immunology, and he's also the chair of the Department of Biochemistry there. He's a world expert on innate immunity and inflammation. His main research interests include toll-like receptors, inflammasomes, and also immunometabolism. He's listed in the top 1% of immunologists in the world based on citations per paper. And he's a co-founder of Citrix, a company that he started and aims to develop new medicines for inflammatory diseases. Another company he co-founded, Inflammasome, was recently acquired by Roche. He has won a number of major awards. I'm not going to list them all here. He was awarded the Royal Dublin Society Irish Times Boyle Medal for Excellence or for Scientific Excellence, the Royal Irish Academy Gold Medal for Life Sciences, the Society for Leukocyte Biology, Dolph O. Adams Award, and he's also a member of the Royal Irish Academy, EMBO, and he's a fellow of the Royal Society. He has a passion for communicating science to the public. He has a weekly radio slot on the Pat Kenny show on News Talk. I wish I could listen to it, but what time of the day does it play?
Well, you can tune in, Hussein. You can go online, you know. Usually it's 11 a.m. on a Thursday morning. So
oh, okay. Yeah. Yeah, so 5 a.m. our time.
Yeah.
He has also written a number of books, including Immunology, a scientist's guide to our amazing existence. And he has also written a book called The Great Irish Science Book, a science book for 10 to 12-year-olds. And he recently published a paper called Never Mind the, and then the next word has a couple of asterisks in it, so I'm not going to mention it, but here's the science. So that's the name of his latest book. I have read many of Luke's papers. I have emailed him a couple of times. One thing about Luke's papers, you know, his science is amazing. I've really enjoyed reading his papers over the past few years. But the review papers that he writes are so easy to follow. And I've actually emailed him and I've mentioned to him how much I enjoyed reading his review papers. So thank you for all the great work you are doing in the field, and we look forward to your talk.
Great. Thanks for saying for those very kind words. Yes. I'll just share my screen. Very happy to give a talk. Shame I can't be there in person. We're living in a very strange world, aren't we? Let's face it.
Yeah. Exactly. But before you start, sorry, let me interrupt 1 second. If for the audience, if you have any questions in the chat, please mention that you have a question. At the end of the talk, I'm going to call on your name so you can ask the question. Sorry to interrupt. Go ahead.
No problem. Yeah, I'm very happy to give my talk. I'm very happy to, as I say, go through some of the recent stuff we've done. I'm going to give a bit of an overview as well. And Hussein mentioned inflammasome. I might give you that story because that was a very exciting development. As Hussein has said, I've got a long interest in innate immunity. And I began working on IL-1 at Cytokine back in 1985. Can you believe it? It seems like the dark ages now, before email was even invented. In those days, we used to go to the library. Do you remember, older people? But then IL-1 was one of the first big cytokines, I guess. I worked on TNF in the mid-80s as well. And as you all would know, TNF became a massive target in the pharma sector. And then I moved more towards, I guess, what drives cytokines. That became the next focus. And I've got into TLRs. I was lucky enough early on to work on TLR4 and TLR9 and discovered some of the signaling pathways. That led me to inflammasomes because obviously the NLRs are part of the same family, and NLRP3 became a big focus, as I'll mention. And then I suppose about 10, 12 years ago now, for some still strange reason, I got into metabolism and metabolic change in macrophages during, TLR signaling initially. That happened because I'd sent a paper to Nature Immunology, it was, on an amyloid factor driving IL-1 production through NLRP3, as I'll explain. And a referee said, but glucose drives IL-1. And that had been shown back in the 80s, actually, the high glucose was a driver of IL-1 production. And that they made me do an experiment by blocking glycolysis, the referee did. And we saw a profound block in IL-1 by blocking glycolysis. And that got my imagination. So what does that mean? Yeah. And then my lab then began to shift more into metabolic change. in macrophages mainly as a driver of cytokines, and gave rise to all kinds of strange discoveries, as I'll explain, and how very specific metabolites actually can plug into cytokine production in various ways. And then the work in NLRP3 gave rise to an effort-defined inhibitors, and that gave rise to our company Inflammasome. So I'll give you some of those as we go along, I guess. But I call my talk Adventures in Inflammation Research, because from the very get-go, for me, it was always about trying to unravel the complex signaling pathways that underlie the inflammatory process at its most fundamental. And strangely, back in the 80s, if you worked on inflammation, you weren't really seen as an immunologist, you know. We were seen as left field people because we didn't work on adaptive immunity necessarily. We were mainly working on, you know, inflammatory pathways, things like prostaglandins, which weren't seen as important for immunity at all. That changed because of innate immunity. That kind of brought inflammation back into the immune world, I suppose. And the innate immunity explosion that began in the mid-90s really made inflammation a key part of immunology again, kind of. You may be familiar with some of the history of what Polly Matzinger called the dirty little secret of vaccines, for instance, or Charlie Janeway's notion of danger and Polly. I mean, they were kind of immunologists who led some of this, I guess, rediscovery of inflammatory processes. So it's funny when you use the word inflammation. It's now seen, of course, as I say, as a key part of the immune response. But I think what's important to start off with is what many of us are trying to achieve clinically in our research and how it translates. As anybody listening in will know, if you take a disease like RA or IBD or MS, there are, of course, breakthroughs in the last 20 odd years, which mainly mean targeting cytokines. But you reach what's called an efficacy ceiling, and some patients don't respond at all. Some respond very well and then stop responding. So there's always this idea that we need to do better, even though the VISTA improved greatly for rheumatoid. It's still not a fully sort of solved disease in many ways, and IBD is still a big problem. So we've got a bit of a way to go with getting really powerful therapeutics for these inflammatory conditions. And like the therapies will maybe get you to say 50% response, and that will be things that target cytokines like TNF, or maybe T cell therapies like abatacept or rituximab. The goal is to breach that ceiling. And what can we do there? And kind of many of us have realized, if we keep working on the same pathways, and that might be a T cell process or a B cell process, you won't beat these therapies anyway, because rituximab depletes 99% of B cells. Or we know TNF is a key cytokine that drives much of this, and there's no point sort of reinventing the wheel. We need to go beyond those standard targets, and that's where things like metabolism come in, as I would explain, and indeed cell death pathways have got more interesting, and things like NLRP3 drive certain cell death processes, and they're the two main things my lab actually has shifted into, and I'm going to give you some of the stories around those two. And if I start with NLRP3, I mean, it's a striking business. This was discovered by Jörg Chopp, for example, in Lausanne. It was also discovered by a guy called Hal Hoffman, who had mapped a gene variant that drives a very rare disease called Muckle-Wells syndrome. When he uncovers this gene, names it cryopyrin, Jörg Chopp calls it NLRP3, and it joined the TLRs. So my lab was kind of poised to work on this in a way, because they're all in the same big family, really. They're all innate immune receptors that have similar domains, actually. So you know, NLRs have leucine-rich repeats, just like the TLRs. And what NLRP3 did specifically, though, was it was the kind of missing phagocytic receptor, if you like. So it was known for a long time that if a macrophage engulfs some particle, it will drive IL-1 production, and NLRP3 turned out to be the sensor of phagocytosed material. So it will sense things like uric acid crystals. It'll sense beta amyloid, for instance, in the brain, or cholesterol crystals in athero, or in our work was IAPP, this amyloid factor in type 2 diabetes. And what was amazing was it was described by Jörg in 2001. And by the time you get to sort of 2011, 2012, there was evidence for NLRP3 in all these different diseases. Now, these were all macrophage driven. They all involved the macrophage trying to phagocytose some material in the tissues, driving NLRP3 to drive IL-1 and driving chronic inflammation. And suddenly the notion then, now this is about maybe 8-9 years ago, the notion that this single pathway could drive things like COPD or asthma or psoriasis or Alzheimer's or Parkinson's or gout, say, or IBD, certain types of cancers as well, which are known to be macrophage driven to some extent. Mesothelioma is a good example there, or indeed, you know, diabetes or athero. all driven by one pathway, the evidence began to grow. Now, it seemed unlikely that a single major innate sensor would be implicated in all these different pathologies. But the evidence grew and grew. And the evidence was based on, say, overexpression in disease or models of disease, knockouts being protected, say, and even some genetics, especially in cancer, interestingly. There's a few papers on NLRP3 mutation there. So this picture begins to emerge of a central pathway driving another pathology. And in my view, then, what happened quite quickly was, and again, you could have made this slide about 3 or 4 years ago, I guess, NLRP3 joins 3 other pathways, which really were well validated and lay at the heart of autoimmune or autoinflammatory diseases. And these are the Jak-STAT pathways. We were discussing that a few minutes ago with John O'Shea's pioneering work, and that will drive gene expression and will drive autoimmunity. The CGAS-STING pathway gets unraveled by people like James Chen. That is sensing DNA in the wrong place, you might say. It could be mitochondrial DNA, it could be microbial DNA driving, say, type 1 interferons, again provoking autoimmunity. And then the RIP kinases there on the TNF pathway as one example, driving its cell death process. And that joined NLRP3 as a sensor of noxious stimuli in the broadest sense. And the output wasn't just IL-1, it was IL-18, a second important cytokine, and gasdermins that they get first described, I suppose, about 6-7 years ago now by Vishva Dixit in Genentech. That will drive pyroptosis, a type of inflammatory cell death, and that will drive autoinflammation. So in some ways, and this is a broad spectrum slide in a way, I think we're getting very close now. I mean, some of the key pathways that drive autoimmune and autoinflammatory diseases, and it's a striking business. Now, of course, what this means is everyone wants inhibitors. And as we mentioned, Pfizer were the first to go after JAK inhibitors. Xeljanz is now approved for rheumatoid. Every company is looking for CGAS inhibitors now. And one disease, there'd be lupus, for instance. There's evidence for aberrant DNA sensing in lupus, driving autoimmunity. NLRP3 inhibitors become a focus, and then RIPK inhibitors is an example, and several of those are in development. So here we have small molecules now that will be orally active, that could either replace biologics or do better than biologics or combine them with biologics. So suddenly the whole area now is sort of become, I guess, more tractable. And remember, the goal here is to develop small molecule inhibitors that will breach this efficacy ceiling, and you'll see more efficacy than the biologics is one notion here. And here's the thing that struck me back in January. I got this Torreya report. It's a kind of a consultancy type document for industry assessing the biotech sector. And it really struck me that this report lists, now this is a tour de force. All of the drugs on this slide are either approved for inflammatory or autoimmune diseases, like there's Xeljanz, there's Actemra, or in clinical trials, and many different drugs are in development. But this analysis homed in on the IL-1 pathway and NLRP3 as being especially exciting with all these inhibitors in development. And it also said all these JAK inhibitors, there's many JAK inhibitors now being tested in all kinds of different systems. So the Jak-STAT pathway and the NLRP3 IL-1 pathway became sort of very prominent for this analysis. And it's a very interesting report in that regard. Now, there's obviously more targets out there than these. But again, it emphasizes the breadth of this now and how many drugs are in development. And our compound, I mean, how we get into this was, I began searching for NLRP3 inhibitors, or it would have been probably in 2012. And we came across a compound that Pfizer had discovered, that Pfizer called it CRID3 for cytokine response inhibitor drug 3. They found it in a phenotypic screen in macrophages to block IL-1 production. And they got a very potent inhibitor, Chris Gable finds this really potent small molecule, nanomolar potency. And Pfizer developed that up to a point. They even go into a trial in rheumatoid. They get some efficacy, but the compound had liabilities, and they drop it. Now, I knew about that compound, and I wondered, maybe that's an NLRP3 inhibitor. And again, it was just sort of a lucky guess in a way. I just said, let's give it a go. And lo and behold, what Pfizer had found turned out to be a highly potent, highly selective NLRP3 inhibitor. We name it MCC 950 for various reasons because it got a bit confusing, but that's the same compound. And we published a paper on this in 2015 saying it's a very selective NLRP3 inhibitor. It blocks NLRP3 in different contexts. We gave that compound out to over 80 labs who asked us for it, and all this data came back. And again, this is a striking business. That single small molecule inhibitor of NLRP3 worked in animal models of all these diseases, Alzheimer's, Parkinson's, ALS. You get into things like steroid-resistant asthma, pain in MS, for instance. MDS is in there, monodiastolic syndrome. And then athero, NASH, autoimmune models like MS, EAE was the example there, colitis, psoriasis, and even some infectious diseases where you have sort of a septic type response. Now, again, it's a striking business that a single small molecule could work in all these different models. The reason being the macrophage is front and center here. NLRP3 is implicated, and lo and behold, this presents a real prospect. Now, what we did next was we then made derivatives of this compound, because obviously Pfizer had dropped it. They'd not much IP around it, strangely. And Matt Cooper, my key collaborator here, he's a co-founder of Inflammasome. We form a company, me and Matt. We make derivatives. We end up with two very potent selective inhibitors that are even better than MCC and then CRID3. And then Roche, very nicely, as Hussein mentioned, they've acquired our company. Now, Roche will embark on 6 clinical trials, they've said, with our molecules in some of these indications. They haven't disclosed what they are yet, but you can kind of guess them. So now it's a thrill for me, because the molecule we showed was an NLRP3 inhibitor, and we developed derivatives, and now Roche will now really test in the clinic to see if this promise comes true. And it's a striking business, and you never know. It may well end up that this NLRP3 pathway, I'll go back to it here again, turns out to be key for many of these pathologies, and we'll see now in the clinic if this is true. Now remember, all these other inhibitors are in development. There's a big effort. That slide there with all these different compounds tells you that. There's many options. Here's our compounds. They've been renamed Roche compounds, of course. One is called Inzomalid, and the other is called Somilix. They're our NLRP3 inhibitors now in clinical development in Roche. So it's a very exciting prospect. We'll see what happens. Now, the other part is, I mean, we work on NLRP3, we all that effort, and we did basic research on it, found some inhibitors, and now we'll see if Roche will make the dream come true, I suppose, for us. There's at least 3 other companies were found on the back of our paper, actually. All the big pharma realize this is a druggable target. You can make small molecules now. There's about 7 NLRP3 inhibitors now in development, so we'll see what happens and see who gets there first. If it's us, I'll be happy. If it's not, well, at least we lit a fire that got the thing going, I suppose, so we'll see what happens. Now, in the meantime, my lab is working more and more on metabolic change and inflammatory signaling pathways in many ways because of our work on IL-1 initially. And again, this slide captures this. It turns out that the mitochondria have gotten more and more interesting in terms of generating signals to drive inflammation in the broadest sense and to drive cell death pathways. This begins, of course, with Xiaodong Wang's pioneering work on cytochrome C release from mitochondria, driving apoptosis. So we knew for about 25 years that there were signals coming out of mitochondria to plug into quite specific pathways. I mean, that cytochrome C pathway is very, very specific with the APAPs and the caspases, But more and more evidence emerges of mitochondrial change driving downstream signals. So with the TNF pathway, we know mitochondrial ROS, reactive oxygen, could drive TNF signaling processes and drive cell death. We know mitochondrial DNA can be sensed by CGAS, and that can happen in autoimmunity. And then for the NLRP3 inflammasome, mitochondrial ROS, again, was shown to be a signal for NLRP3. It's amazing. NLRP3 actually goes to the mitochondria to be activated. And evolutionarily, it's very interesting that cells are using the mitochondria as a kind of a signaling hub, I suppose, to provoke inflammation. And in many ways, the phrase I use is these are ancestral prokaryotes, remember, and we tolerate them in our cells until something goes wrong. And now they begin to drive an immune response. They begin to drive kind of an innate signaling process that might drive downstream signaling events. So conceptually, it's a fascination that the mitochondria now become this very important driver, I suppose, these downstream events. And I'm going to give you 2 stories that we've done a lot of work on the past few years from our lab on 2 mitochondrial metabolites that surprisingly plug into quite specific signaling pathways in the macrophage from Krebs cycle, because they're both from Krebs cycle. And one is succinate, and there was Itaconate, and I'm going to give you those 2 now. And in fact, just to remind you, in case you've forgotten how important Krebs cycle is, now, immediately, anybody who's done medicine gets scared now because they learned this cycle, didn't they, in medical school? Learn it again because it's extremely important for many other things. And you would have learned it as a pathway, a cycle that drives electron transport to drive ATP production, a triumph of biochemistry, of course. But remember, it is the central metabolic hub of life because it drives amino acid synthesis for proteins. It can also drive purines and pyrimidines for nucleotide synthesis, and you can make lipids from citrates. So it's always known as the central hub of all metabolism. But now I'm going to home in on two parts, itaconate there and succinate. And now you may remember this, or you learn it and you forget it and you learn it again, you see. But that's how we all had to learn this cycle, didn't we? But these two metabolites, succinate and itaconate, are what I'm going to talk about. Now, I'm not going to give you much more on succinate because we've published a lot on it. The bottom line is succinate is a pro-inflammatory metabolite. It will drive reactive oxygen species from complex 1 in the mitochondrial electron transport chain. That will drive reactive oxygen, and that would in turn provoke things like HIF. And HIF can drive IL-1. We published a lot on that 2 or 3 years ago. We did read a few papers on that. So what succinate, if you like, is a pro-inflammatory signal. This one, itaconate, you may have heard less about that. That is coming off Krebs cycle. An enzyme called IRG1 metabolizes itaconate from Krebs into itaconate. And guess what? I'll give you the punchline at the start. Itaconate's anti-inflammatory. So it looks as if you've got Krebs driving succinate to be pro-inflammatory, and then it'll divert away from Krebs into itaconate to block inflammation. And there's a real yin and yang here, we think, between these two Krebs cycle metabolites. Now, again, I can't emphasize enough how surprising this is at one level. Krebs being boring and old-fashioned, suddenly being very specific in terms of having effects on quite specific downstream inflammatory pathways to provoke inflammation through succinate or to limit it through itaconate. Okay, that's the kind of overall concept. Now, there has been an explosion of information on itaconate. And again, if you go back to 2011, there's like 6 papers on this metabolite in immunity, very sort of underexplored. But lately, all these papers have come out. And what's striking is there's papers that are having effects on pathogens. So there's antimicrobial effects. There's stuff on salmonella, paper in Science about 6 months ago on itaconate and anti-salmonella, basically blocking metabolic pathways in salmonella. There was lovely stuff from Alice Prince on itaconate and Pseudomonas. There's also stuff on cholera. And then most recently, the dreaded SARS-CoV-2. There are effects on viruses, back to that in a minute. These are direct antimicrobial effects of this metabolite. Equally, there's a literature on itaconate having effects on the host in various ways. And there were 2 or 3 papers on this about 3 or 4 years ago on itaconate limiting succinate, which was interesting from our point of view, kind of built on our succinate data and blocking the effects of succinate. That was good. And then since then, a few more papers on itaconate affecting host defense pathways and affecting inflammation. There's a fair few papers now have emerged in the past, 2 years on this. And the effect on viruses is intriguing. Now, we're going to have more on this a bit later. We had a paper in 2018 on this as part of our first paper on itaconate. It can affect type 1 interferon production in itaconate. Now, remember, it's a Krebs cycle derived metabolite plugging into type 1 interferons. There was also evidence for effects on Zika virus, effects on influenza, and effects on VSV. So there's this literature on itaconate affecting viruses. Back to that a little bit later. And how my lab got into it was very simple. Back in 2016, we did a big metabolomic screen of LPS on macrophages. Now, we often use LPS as a kind of a hammer blow to macrophages. It's a powerful macrophage inflammatory stimulus, goes through TLR4. We worked on that pathway for years. Here's LPS causing a 6 log increase in itaconate. Now, we'd seen that and didn't know what it meant because it's a really obscure metabolite. But because the magnitude was so profound, I said, let's work on that. That's got to be important, you know? So my lab then begins to work on this metabolite. And we asked the question, what is this metabolite doing? It's massively abundant in the inflammatory macrophage. What's it doing? Long story short, it's anti-inflammatory. And we've loads of evidence on this. And I'm going to show you some of it. But that's to keep your head on an anti-inflammatory pathway. And then most recently, there's been 2 papers I want to pick out for you, which confirms our conclusion. Now, other labs were saying the same thing. It wasn't just that it was anti-inflammatory. But we had a very strong sort of hypothesis here. And look at this, a recent paper on rheumatoid, where they did a metabolomic screen. We're in the world now, multi-omics, as you all probably know. And look at this, they could correlate itaconate with the DAS score. Now, any rheumatologists happen to be listening, the DAS is used as a bog standard scoring system. And look at this, the higher itaconate was, the lower the DAS score. See, here's the DAS, there's itaconate, you know, inverse correlation. In other words, if you have low itaconate, you have a high DAS score, and it's inverse to CRP. And that was correlation suggesting perhaps it was anti-inflammatory, because the lower it is, the higher the inflammatory score. And in this paper, came out back in August now, on COVID-19, it was an unbiased metabolomic screen on COVID patients. And again, they got, look at this, the macrophage specific metabolite derived from itaconate was progressively reduced with COVID-19 severity. So again, an inverse relationship with COVID severity. And then, you know, that was more evidence to suggest an anti-inflammatory effect. So, and that's just a little snapshot of a recent literature. Now, our lab had worked on its anti-inflammatory effects. The big question for us is, what's the mechanism? And again, to cut a long story short, it reacts with cysteines in target proteins. So here's a metabolite being made in a macrophage, cysteine reactive, and when it modifies cysteines, it inactivates the proteins it modifies. And these proteins are pro-inflammatory. In our paper, we listed 30 proteins that itaconate was modifying. This was inspired by fumarate, another Krebs cycle intermediate, which is not well known to modify cysteines on proteins and modulate them. And we found itaconate was a very good cysteine modifier. Things like LDH, for example, that would block glycolysis, and glycolysis is pro-inflammatory. Guilt is in dendritic cells. It's an antigen presentation, for example. So we found a whole set of proteins being modified by itaconate, and that's part of its mechanism. And in fact, one we found was NRF2 activation. It modifies KEAP1 to drive NRF2, and that's anti-inflammatory. So we published on this in 2018, and its mechanism then is partly to do with cysteine modification. Back to that a bit later, actually, as a kind of a therapeutic concept. And then most recently, we found other targets, and I'm going to give you these two stories now. And guess what? It modifies NLRP3, and that kind of unites two parts in my lab now very nicely came together. And it turns out itaconate is an endogenous NLRP3 inhibitor and inhibits it, okay? And that's partly why it's anti-inflammatory. I'll give you that story. Second story is the Jak1. It turns out itaconate is a Jak1 inhibitor, as I explained. And then we've got data with COVID, which I'll come back to, that was inspired by that inverse correlation. So first of all, NLRP3. Now, what inspired this was there was evidence for NRF2 blocking NLRP3. And it was doing it partly through suppressing reactive oxygen, because NRF2 will drive various antioxidant pathways, and they can limit ROS, and that could then impact them. That's what inspired these experiments. We thought that itaconate would be an NLRP3 inhibitor through NRF2. That wasn't the case in the end. And it turns out we got more and more evidence of it being a direct inhibitor. Now, we use derivatives sometimes. This is 4-octyl itaconate. We're using that as a way to get itaconate to cross the membrane, because itaconate is a bit electrophilic. So, and it mimics itaconate. There are differences, I will say that. But we treat 4-OY or derivative as a kind of proto-drug, because we're interested in making medicinal agents from itaconate. And so some of the data is based on 4-OY. But look at this. It blocks IL-1. So there's ATP driving NLRP3. Nice dose response to itaconate. You can use nigericin. Any aficionados will know this. That will drive NLRP3, again being blocked by itaconate. And we mentioned IL-18, because that's an output from NLRP3. Profound inhibition of IL-18 with itaconate. So this all suggested it was blocking NLRP3. And more evidence, we took IRG1 knockouts. Now, the enzyme that makes itaconate is called IRG1. That's the enzyme that takes itaconate and makes itaconate. We took knockouts and we got a boost in IL-1, which was nice, because that means endogenous itaconate wasn't being made here, because IRG1 is knocked out, and you get a boost in IL-1. That was more evidence. We got nice evidence for P20 here, for caspase 1, for example. And then we took some cells. I'm only giving you tiny bits of the data. We have loads of data on this. This was pretty compelling. We took patient samples from patients with this rare autoinflammatory disease called CAPS, Muckle-Wells syndrome. Mentioned it earlier. These patients have an activating mutation in NLRP3. Their blood, you get PBMCs, they're hyperactive in response to LPS, because NLRP3 is mutated. And you can use ex vivo. Ex vivo, we could block all the IL-1 in CAPS patient samples, and that was great. There's our CRID3 MCC 950 compound blocking it anyway. So again, that was good evidence. All that's wrong with these cells is they've got overactive NLRP3, and now we're blocking with itaconate. That was nice. The question was, how does it do this? And of course, we counter-screened against AIM2, NLRP3. They weren't inhibited, so it wasn't caspase 1. We could rule that out. We went direct for NLRP3 and NEX7, which is in the complex. And again, we got very nice evidence of NLRP3 modification. And itaconate modifies cysteine 548 and NLRP3. And we think that's the mechanism. It's very hard to confirm that definitively because you got to make mutants and it's not easy. But we think that's the mechanism. So itaconate is a direct NLRP3 inhibitor. And we got the cover of cell metabolism to boast. Us Irish are very modest, as you know. Itaconate targets NLRP3. On the cover, they put all the inflammasomes. Isn't that nice? So here we have a really interesting finding that there's an endogenous metabolite modifying NLRP3 to limit inflammation. And it adds to our evidence that itaconate's anti-inflammatory in the broadest sense. Now, the second target we got is Jak1. Now, as I mentioned, the JAKs are a very important class of drug targets for the industry. Anyway, we think we've come across a very interesting Jak1 inhibitor. Now, what made us look for Jak1? Well, again, it was a separate project in my lab. We knew early on, and this was shown by other labs, only M1 macrophages make itaconate. It's never made by M2s. They don't have the IRG1 enzyme. So it's a real marker of the inflammatory macrophage. And that was interesting anyway. And of course, the reason is a negative feedback loop, by the way. Why do M1s make it? It's to feed back and limit inflammation. You know, that was the finding anyway. But we wondered, you know, what would this mean for M2 macrophages? There was evidence M1s can release it. So they secrete itaconate, and other labs had shown that. And then about 2 years ago, a very nice paper showing M2 macrophages can take up itaconate. They've got the transporter. So we wondered, well, maybe M1s would release it to limit M2s. Now remember, M2 macrophages, they're called alternative macrophages. They're inclined to be anti-inflammatory and tissue, you know, they resolve inflammation. They respond to IL-4. So we wondered, maybe M1s turn them off to provoke more inflammation. And we began to wonder then, would itaconate impact on M2s? And then, you know, what was striking about this was we gave M2 macrophages our derivative, our 4OI, and it was a profound M2 inhibitor. So here's IL-4, which we use as our stimulus for M2s, driving ARG1, blocked by itaconate. There's FIS1 is blocked, MRC1. All the M2 markers were suppressed by the 4OI, our derivative. And then we turn to IL-13. That's another great M2 driver. And again, itaconate could block IL-13-driven responses. We also tried the real molecule, itaconate itself here, and that could block as well. Not as effectively as the derivative because it can't cross the membrane. The transporter is there, but 4OI gets in more readily. But the real itaconate can also block IL-13 action. Here, there's p-part gamma being blocked by the derivative and by itaconate. And then, of course, because IL-4 and IL-13 were blocked, we said, well, what's common to those signaling pathways? And they both go through Jak1 and STAT6. They share that signal. So we wondered, is Jak1 being targeted or STAT6? So we measure these as readouts. And very nicely, you can block phospho-STAT6 activation. Now remember, IL-4 and IL-13 will drive phosphorylation of STAT6. And look at this, there's itaconate itself blocking phospho-STAT6. There's the 4OI blocking that. So it's blocking STAT6 phosphorylation. Next, we looked at phospho-Jak1 because, again, anybody who knows these pathways, the JAKs auto-phosphorylate as part of the signaling pathway, and you can measure phosphorylation. And look at this, very nice inhibition of phospho-Jak1 and by itaconate and 4OI, and that was good. So now we're getting close to the action. It's Jak1 that seems to be being targeted here. And then, you know, what was nice then was a lab in China, Chu Wang, they're a very good sort of cysteine modification lab. They had a paper in JAKs, interesting name, JAKs, talking about Jak here, identifying Jak1 as a target for itaconate. That was an unbiased screen that Chu Wang had done, and he says Jak1 might be modified. I contact Chu, and he's very helpful to us. He's got a great probe called the ITalk probe, which is a modified itaconate, and you can measure using chemoproteomics modification of proteins. We got him to send us that probe, and we got very nice modification. This is direct modification of Jak1 by 4OI or derivative. You can put it down and blot. It's a simple enough assay. That's incorporation of itaconate into Jak1. And then no other JAKs were modified. We counter screen many JAKs. And lo and behold, literally an hour ago, Hussein, I'm going to show you the data. He sent me the mass spec data. 4 cysteines in Jak1 are modified by itaconate. And these are very interesting cysteines. They're in the active site. So again, we've evidence now direct modification of Jak1 by itaconate. And it's another target for this very interesting metabolite. Now, we also went in vivo because it's, I mean, you got to be careful with metabolic work. You got to go in vivo to really confirm things because cells in culture are in very weird media. Went in vivo. You can challenge mice with IL-4 directly. It's an IL-4 antibody complex that will bring out M2s into the peritoneum. You make PECs in the mice actually, and then you can measure gene expression and look at this in vivo. Then now obviously you measure this ex vivo, but you're isolating the macrophages. ARG1 was inhibited, FIS1, YM1 in vivo. That was giving mice now 4OI in vivo. We also looked at phosphostat6 in the macrophages taken from the in vivo treated mice. And again, phosphostat6 was down, as was phospho-Jak1 in the macrophages. So we can block this pathway in vivo. It means itaconate's active in vivo to block M1 macrophage function. And so what this means in a nutshell then is itaconate is a Jak1 modifier. Now, it's especially the case with the derivative, I must say. The endogenous stuff, we've limited evidence for that. It's very hard to show this, that M1s talk to M2s in vivo. It's hard to prove that. But at a minimum, what we found here is another Jak1 inhibitor based on itaconate. Now, we also did stuff in interferon beta signaling, because that goes through Jak1 as well. And we showed effects on Th2 cells. We can put IL-4 on a Th2 cell. They were blocked as well. So we think we've discovered a new Jak1 inhibitor, basically, based on itaconate. Now, the cysteine modification is interesting, because there is a Jak3 inhibitor called Ritlecitinib that works by modifying cysteines in the active site. So others had shown cysteine targeting by Jak inhibitors already. We've just shown this now with a potential endogenous mechanism. And secondly, another way to block cysteines, and it's specific for Jak1. That's the important thing. We found a Jak1 specific inhibitor. Now, lastly, I'm going to talk about our COVID work, because I can't resist it. We're all obsessed with this virus, aren't we? And my lab, like many labs, began to work on COVID. I guess about a year ago now, we switched over into COVID work. And some of the motivation, why would we do this? It's a very competitive area, let's face it. Many labs are doing it. We did it because our molecules, our obsessions, if you like, were implicated in COVID-19. And there was a good literature now emerging on NLRP3. Now, clearly, once COVID goes systemic and once you get towards a severe end of the spectrum, loads of macrophages, you know, it's obviously a macrophage disease. It's a bit like ARDS, in case, you know, I guess there'll be people listening who work on those things, maybe. Slightly different to ARDS, though. It's not like a simple respiratory disease like you give it LPS, for instance. But NLRP3 was there. And this is a review that was just done back in June, actually. There is NLRP3, sensing COVID, driving IL-1, driving cell death. So there was evidence for this. And in fact, there was a paper in JAXMed about 3 months ago now, Dario Zamboni. He took samples from patients with COVID and very severe patients, big activation of NLRP3. You can measure it in PBMCs, that green speck. You can measure it in the lung tissue as well. So there was evidence for NLRP3. And then we ourselves, probably 9 months ago now, we measured IL-18 in peripheral PBM, in serum samples from patients. We took severe patients versus non-severe. IL-18 was elevated. Now, others had seen this as well. So IL-18, in fact, could be a good biomarker for severe COVID, and we can confirm that ourselves. There was evidence of IL-18 in the inflammasome, I guess. And then remember I mentioned the inverse correlation between itaconate and COVID-19. That got me thinking, you know, maybe there's less itaconate in severe disease. That means elevated NLRP3, driving that IL-18 signature. So my lab begins to wonder, let's look at itaconate and NLRP3 in the context of COVID. I'm going to show you some data now. We began to work on this. The other thing that was nice for us was there was a paper in Cell Metabolism back in September now, where they showed that SARS-CoV-2 will drive succinate. Now, remember I mentioned succinate being pro-inflammatory. That was all LPS. We mainly use gram-negative bacteria and LPS to drive succinate. Nice paper, repeated all our work actually using SARS-CoV-2. Succinate, HIF, cytokines. So again, that was all relevant to my lab, I guess. And we began then to consider this as an area for our lab to work on. And then remember I told you how there was the itaconate had antiviral effects. And the Zika paper had this in as a little diagram. There's itaconate being sprayed on Zika virus. That got me thinking, oh, itaconate could be interesting in the context of COVID-19. And then remember, there was already evidence that things like dexamethasone, you may be familiar with this, was working in COVID. So the anti-inflammatory properties of itaconate then became all the more relevant, I guess. So we got some data. And again, Martin van Hemert, he's in Leiden. He had a very nice human lung epithelial cell model for COVID. You infect with SARS-CoV-2. It's cytopathic. It will kill those epithelial cells. We put itaconate on those cells and we can stop the cell death. Here we see inhibition now, dose response of the cytopathic effect of SARS-CoV-2 on epithelial cells. And we could suppress replication. I mean, here's the RDRP gene being blocked. There's the E gene in COVID-2 being blocked in supernatants and in cell lysates. So we think what this means is a direct antiviral effect. And as I mentioned, there was evidence for itaconate having a direct anti-Zika effect. So that suggests it's directly antiviral. And then look what have it, Christian Holm in Denmark, in Aarhus, scoops us. He had a paper on 4OI, or derivative, blocking SARS-CoV-2 through NRF2, we think. So there's evidence of an antiviral effect here of itaconate and derivatives against COVID. But then we also did one last thing I'll show you. We went in vivo with collaborators Lawrence Liesenborg in Leuven, and they've got the classic hamster model of COVID-2, and they infect the hamsters with SARS-CoV-2, and they get lung pathology. And it's a complicated scoring system. They measure things like bronchiolitis and edema, and you get a score. And you know, these are hamsters, you infect them and you get an increase in inflammation in the broadest sense. We gave those hamsters our derivative in vivo, and look at this, we could suppress the lung inflammation. And we think this could be a combination of an antiviral effect to limit replication, but I bet it's also being anti-inflammatory. It could be targeting NLRP3 in this situation, or other targets we identified for itaconate. So again, we're now following up on this, obviously. And my company, Citrix, that Hussein mentioned, we're developing itaconate derivatives now to go after inflammation in the broadest sense, but equally it could be a prospect. I think my big idea here would be is if it's true that itaconate is inverse, you know, in other words, it gets depleted for some unknown reason, you could replenish it and have a therapeutic effect on a severe respiratory syndrome such as COVID-19 is. And again, remember, our work will go beyond COVID into other respiratory situations, we hope. So that's our COVID work that we're very intrigued by. Now, I'm going to finish with a couple of sort of bigger picture things in a sense. So I guess you've got the picture now, I hope, that this metabolite, itaconate, it's made by inflammatory macrophages. It now begins to react with key target cysteines on target proteins and begins to have anti-inflammatory effects. That's the overall concept. Now, that concept was there for fumarate in the past, by the way, of cysteine modification. And is this any way sort of valid, I suppose? Well, I think it is, because there's a growing literature now on cysteine modification to modulate downstream pathways, especially in the inflammatory process. And we've got two endogenous metabolites, fumarate and itaconate, modifying these cysteines and inflammatory proteins to decrease inflammation and maybe drive resolution. And there's loads of targets, GAPDH in glycolysis. We've shown LDH. There was a paper on gasdermins being modified by fumarate in Science by one of my former trainees, Kate Fitzgerald. That was quite nice. So there's a list of proteins here that are interesting, of course there is, and we would put NLRP3 on the list, because obviously from our data now, NLRP3, Jak1, and then we've got what's very interesting, I think, is, and really validates this, dimethyl fumarate is a drug used by Biogen own that is called Tecfidera. That's used widely in MS. That modifies cysteines. Its mechanism is cysteine modification. I think that is just a lucky break. It is mimicking an endogenous pathway with fumarate, you know, that modifies cysteines. A very interesting paper about 6 months ago on disulfiram. This was a random screen of compounds to block pyroptosis, that type of cell, that HOU finds disulfiram modifying gasdermins. So again, these two compounds in humans already to treat various diseases, if you like, are modifying cysteines for therapeutic effect. And I'm putting my 4OI, my derivative, on the drug side, because it mimics itaconate to some extent, but has separate properties as well. And then we're very keen on pursuing 4OI as a possible therapeutic, or compounds based on this to modify cysteines. And remember, I think what's happened is these were discovered empirically, DMF and disulfiram. And maybe they're working because they're mimicking an endogenous pathway through modification of cysteines by Krebs cycle metabolites. And now we see an anti-inflammatory effect. Remember I said already there is evidence already for things like Ritlecitinib, a Jak3 inhibitor, working through cysteine modification. And there's a Bruton's tyrosine kinase inhibitor called Brutinib, which is approved for autoimmune diseases in certain contexts. Again, that's a cysteine modifier. So in other words, it's possible then to design small molecules to modify cysteines to have an anti-inflammatory effect. The big question is specificity and How many are modified? The modification by itaconate is a brand new modification. We were first to describe it. It's called dicarboxyprobylation. Fumarate succinates these cysteines. So again, we're emerging now into quite a complicated work where cysteine modification could be an anti-inflammatory strategy, which kind of is used endogenously, but could also be used by therapeutics. And of course, the next phase of our work now is how many substrates are there? What determines that specificity? And can we get therapeutics that could have potential then as ways of limiting inflammation and indeed autoimmunity? And that's where that work is going to go next, I suppose. We're on that journey. And remember, if NLRP3 inhibitors make it, well, I'm out of a job. That cracks all those diseases. But many things fail, you know, as you all know. I've been at this long enough now. These give us more options to break that efficacy ceiling, being my overall theme here, remember. And remember that slide, NLRP3 and metabolism could be ways to break that ceiling and get better therapeutics for these diseases is the overall idea here that we're now pursuing. And I will finish with that vista and thank the people in the labs. Alex did all the work on NLRP3, there he is in the back. Mara, she did all the work on Jak1 inhibition. Hauke Weiss helped with some of the COVID-19 work. There's Dylan, he's holding up some itaconate. That was in our lab a couple of years ago. So grateful that the people who did all the work. And then very important collaborators, because it wouldn't be possible without them. Mike Murphy in Cambridge is my overarching mitochondrial guru who helps me. Chu Wang, I've mentioned him, and Jialing Zhang, I showed some of their data on the Jak1 modification. Richard Hardy makes the derivatives. He's the man making our drug-like molecules. Great chemist there. Roman Fisher in Oxford, he is an expert on cysteine modification. He helped identify some of the cysteines I mentioned. I mentioned Martine and Clarice in Leiden on the COVID epithelial work, and then Lawrence Leisenberg in Leuven helped with the in vivo work. And the funding was from the European Research Council, the Wellcome Trust, and Science Foundation Ireland. Thank you very much for listening, and I'm very happy to take any questions.
Great, thank you so much, Luke. That was a really interesting, wonderful talk. Let me start off by asking a couple of questions. So first of all, have you done tracing to see what molecule what versus, you know, lactate versus glucose versus glutamine, which one of them produces itaconate?
We have indeed, I say that was in our paper, but that was in the bloody supplement. We tracked it, we were the first to map that. Glutamine and glucose, they feed in. So, and glucose gets as far as citrate. Glutamine goes around Krebs all the way around, and then you get into, right. So the carbons come 50-50, we think, actually. So you need glutamine and glucose metabolism to drive its synthesis. So it was quite straightforward in the end, in a sense, you know. And as you may know, you get a break in Krebs cycle after itaconate, you see. So you get a diversion into itaconate from those two metabolites.
And then it can freely go in between mitochondria and cytoplasm, right?
It can, yeah. Again, it's amazing, I say, how this, well, it wasn't neglected. Nobody had any interest in this metabolite for decades, you know. Yeah. We mapped the dicarboxylate transporter, actually. So it comes out on the same transporter as succinate, and we mapped that as well. We could characterize that. So that was all in our 2018 paper. We were asked, the referees asked for a lot, actually, because not much was known about those questions, you know, so we were able to map it. Our paper really did three things. It described its synthesis. Now, mind you, a guy called Carsten Hiller, he gets the credit for the enzyme IRG1. That was discovered in 1985. Yeah. An interferon-inducible gene, but no function, you know. He showed it was the enzyme, you know, it's the enzyme that makes a decarboxylase. So we characterize that. We characterize the flux, and then we characterize the transporter, and then also all the cysteine modifications. Nobody'd seen that it was modifying cysteine.
It's only producing macrophages or T cells, B cells. Do they also produce it?
It's very restricted. It's in myeloid cells, M1s we think mainly, but you do, neurons make it. That's Zika paper. That was a neuronal model. Neurons can make it. IRG1 is quite restricted. It's in neutrophils. It's not in T cells or B cells. So the main source is probably myeloid cells.
Okay, great. Wonderful. Thank you. There is a question by Rui, Dr. Ma.
Yes. Thank you. It's a really great talk. I have two questions. My first question is how stability of the 401 in vivo. My second one is if 401 inhibit M2 macrophage polarization, I'm wondering if the 401 has some effect on the tumor immunity, like if 401 could inhibit tumor growth by targeting tumor-associated macrophage in vivo. Yeah, that's my question.
That's a lovely question. Right? Nobody's looked at itaconate in the context of tumors at all. It's well worth considering effects on as you say, TAMs or or even MDSCs, for instance, you know, so that's a really good question. We haven't looked at that. Dan McVicker, a guy at the NIH, he's published a paper on a bit on this actually, on macrophage-derived itaconate modulating macrophages in tumors. And it does suggest that it's got anti-tumor effects, interestingly. So, but that's a work in progress. So it's well worth considering that question, to be honest. How stable is 401? It's pretty stable. Yeah, we've done a fair bit on that actually. We've got the PK/PD, for instance, so we can use it in vivo. We've used it in 2 or 3 models. I mean, I showed some of the data on the IL-4C effects there. We also put it into an EAE model. It seems to work in that. We're making better molecules, though. It's not ideal. That octyl group is a bit of, it's slightly toxic, to be honest, the octyl group, you know. So that's not a great thing to use. But again, people are using it in vivo. And we are making other compounds that are probably, itaconate itself is quite stable in vivo, actually. If anything, that's even better than 401 in vivo. So it's always good to use the endogenous molecule anyway, you know, just to make sure. So you can use itaconate itself.
Thank you.
Marty, do you want to ask your question or do you want me to ask it? You want to unmute yourself?
You can ask it if you like. Go ahead.
No, it's okay. Go ahead since you unmuted yourself. Go ahead.
Okay. So my question I was sent was there's a yin-yang type relationship with some anti-inflammatory drug candidates and trials in which IL-10 goes up and TNF-alpha goes down. So is there evidence for a succinate, what do you pronounce it, itaconate link in this process? Yeah. Is there any link in there?
Well, you know, that's a really good question. I'll tell you what, I should have shown my succinate data. When we did our succinate stuff initially, we did a single-cell RNA-seq experiment, and succinate massively repressed IL-10, you know, and massively boosted IL-1 and TNF, and that was interesting. The reason for the boost in IL-1 is HIF dependency, because IL-1 is a HIF dependent gene. We never cracked the IL-10 suppression. There's evidence coming out of a lab in Brazil, Zika Fonseca. He's got the mechanism of IL-10 suppression by succinate. So it's really interesting, isn't it? Now, itaconate doesn't seem to affect IL-10, to be honest. We thought we measured that, of course, and we were trying to see if we could boost it, for instance, you know. We didn't see much evidence for a boost in IL-10 with itaconate, so that doesn't quite hold up. Although in vivo, that's when you're at itaconate, you know, as a sort of a derivative to measure IL-10, you don't see a boost. But on the other hand, there is some evidence of itaconate blocking SDH, interestingly, and that would limit the capacity of succinate to block IL-10. You know what I mean? So there may be an indirect route into IL-10, but we do, I mean, it's probably oversimplistic, but we do see itaconate as being like the IL-10 of the immunometabolic world, by the way, because it's kind of mimicking IL-10 in many ways, you know? Whereas with succinate, that's more like the IL-1 of the inflammatory world. So I think metabolites can have cytokine-like properties.
Okay, thanks.
All right, great. Any other questions? If not, thank you, Luke. Thank you so much. That was a really nice talk. I know it's 6 p.m. your time.
Yeah.
And if we can chat for 15 minutes, I would.
And of course, I'm happy to
so I'll let the rest of the team or group log out. Thank you, everyone, for your, and thanks again to Luke for this wonderful
thanks, everybody, for coming
presentation.
along.